Anandi, working as assistant professor in the Department of Pulses, DNA Coimbatore. Today we are going to see about artificial chromosome construction and its uses. The various artificial chromosomes are bacterial, plasmid, yeast artificial chromosomes, mammalian artificial chromosome, human artificial chromosome, and plant artificial chromosome. Before going to artificial chromosomes, first we will see various terminologies and related tools in related to artificial chromosomes. Clone. This clone is a word that is used differently in different aspects. In agriculture crops, clone is vegetatively propagated asexually produced progeny of an individual that is used as a propagating material. For example, in fodder grasses, in potato and all, we use clones. In tissue culture, homoclones means an individual grown from a single cell of the parent and genetically identical to the parent. In molecular biology, clone is a piece of DNA fragment of interest found inside a cloning vector. So clone is generally, in all these aspects, the common aspect of clone is it will be uniform and it will be alike that of the parent, that is produced from which it is produced. DNA cloning, a method of identifying and purifying a particular DNA fragment of interest from a complex mixture of DNA fragments and then producing large number of fragments, that is clones out of the DNA fragment of interest. So the purpose of cloning is a particular gene can be isolated and protein, enzyme or the RNA function can be identified and mutations can be identified, and again the organisms can be engineered for specific purposes like insulin production, insect resistance, etc. Cloning tools. The different cloning tools are restriction endonucleases, that is the enzyme that cleaves the DNA fragments at specific restriction sites or recognition sites. These enzymes are found in the bacteria or archaea and provide a defense mechanism against the invading viruses. The restriction enzymes recognizes a specific sequence of nucleotides in the double-stranded and cut the DNA. The restriction enzymes can be classified based on the number of bases in its recognition site, 4 bases or 6 bases, based on the number of bases. Many of them are palindromic, that is, the sequence reads same as backwards and forwards. The palindromic recognition sites reads the same on reverse strand as it does in the forward strand also. Both are in same but in different orientation. Both read same in the same orientation. For example, GAT, ATC. These are palindromic sequences. These are some examples of palindromic sequences. There are 4 types of enzymes, type 1, type 2, type 3 and type 4 based on its composition and enzyme cofactor requirements, nature of the target sequences and position of the DNA cleavage site related to the target sequences. Ligase DNA. These are the specific type of enzyme which facilitates in the joining of DNA fragments. After this we saw how the DNA is cleaved or cut using restriction enzymes. In molecular biology, purified DNA ligase is used for DNA cloning, that is to join the DNA molecules together to form a recombinant DNA. The DNA ligase cannot ligate blunt end DNA, whereas it can ligase when the both ends are sticky ends or one end is sticky, and it is commonly used in molecular biology experiments. Ligation occurs in 4 steps. Reorganization of activity site such as nick in DNA segments or Okazaki fragments. Then adenylation, addition of AMP of a lysine residue in the active center of the enzyme. Pyrophosphate is released and the transfer of AMP to the 5' phosphate of the so-called donor. Formation of pyrophosphate bond. Then formation of phosphodiester bond between the 5' phosphate and formation of pyrophosphate bond. Then again it formation in 3' hydroxyl end as the acceptor. Then vector. Vector is a instrument. Vector is a DNA fragment that is used to clone a gene. Its DNA sequence must be attached to some kind of carrier. That kind of carrier is called as vector. It is also made of DNA, and these are the carriers are called as vector. Vector act like a handle for the DNA, and it also contains other tools such as origin of replication and selective marker. Selective marker is a specific sequence that is having antibiotic resistance or a specific sequence that has code for a specific enzyme. Then choice of vector is based on insert size, vector size, restriction size, copy number, cloning efficiency, ability to screen for inserts, etc. Then plasmid as a vector. Plasmids are small circles of double-stranded circular DNA. They are self-replicating by itself. They are extra chromosomal DNA molecules present in the cytoplasm. They are usually found in the bacteria and yeast, and they can be separated from their very large genome. Naturally, plasmids are of length 5,000 to 4 lakh base pairs, but plasmids have been turned into vectors that can hold 10,000 nucleotides base pairs, that is 10 kilo base pairs. Genes in humans are 3 billion base pairs. More than 1.8 billion clones are required to examine an entire human genome. That is, the plasmid size is very small, and we require at least 1.8 billion clones to clone a human genome. That much of genes are there in humans as compared to the plasmids. Then features of plasmids. For any artificial chromosome, the origin of replication, unique restriction site, antibiotic resistance, and small size facilitates the entry into the cell. The other artificial chromosomes can clone large size DNA fragments also based on its insert size. PBR322, an example for plasmid which has generated from the Boyer lab at San Francisco by Bolivar and Rodriguez in 1972. It is one of the first and most widely utilized vectors and foundation of yeast artificial vectors created by Murray and Swag in 1983. The plasmid contains ampicillin and tetracycline resistant genes, then restriction enzyme target fragments for inserting DNA fragments. Steps in cloning. Then phages as vectors. Bacteriophages can also be used as vectors, where the phage mid or the bacteria are used as vectors in this. It can accommodate larger fragments as compared to the plasmids, and they have, in addition to the helper phage, to switch the phage mode of replication, resulting in the packaging of single-stranded DNA in phage heads. Features of bacteriophage. 1/3 of the genome is non-essential and can be replaced by foreign DNA. Hence, 40,000 to 53,000 base pairs can be inserted for packing recombinant DNA only. In plasmids, we saw 10,000 base pairs, but here in bacteriophage, we can pack 4 times more than that of the plasmids. Cosmids as vector. Cosmids, also called as plasmids, are weird mix of plasmids and phages and can hold between 5 to 35 kilo base pairs of DNA. They are artificially constructed cloning vector containing COS gene, that is cohesive end sites of phage lambda, and require viral packaging prior to transfection into bacterial E. coli. The cosmids can be packaged in lambda phage particles for infection into. This permits cloning of large DNA fragments, that is up to 45 base pairs can be cloned into bacterial hosts in plasmid vectors. However, if the insert size is increased by 23 kb, packaging and transformation efficiency is affected, which are critical for library constructions. Then types of cloning vectors. Plasmids, bacteriophages and cosmids. These three we have already seen, and now we will see the artificial chromosomes. The artificial chromosomes are synthetic chromosomes of DNA fragment integrated into the host chromosome and introduced back into the host cells to propagate and can be used to transfer other cells, that is introducing new DNA. The bacterial artificial chromosome, yeast artificial chromosome, human artificial chromosomes, then plasmid or plasmid artificial chromosomes, and plant artificial chromosome, mammalian artificial chromosomes are the various types of artificial chromosomes which we will see in detail in this lecture. First is bacterial artificial chromosomes. This was first developed by Sisuwa in 1992, and it has a single large copy of plasmid and contains inserts of less than 300 kb. It contains F plasmid origin of replication, and this F plasmid gene controls the plasmid replication and plasmid copy number also. Then uses of bacterial artificial chromosomes is that they are used in constructing large insert genomic libraries, and fragments up to 300 kb can be cloned to bacterial artificial chromosomes. It can be used in genome projects where the number of kilobase pairs is more. That is, here we have 300 kilobase pairs, whereas it was only 40 kilobase pairs in case of bacteriophages, and even less than that in case of plasmids. So bacterial artificial chromosomes was utilized in human genome project to detect genes and large sequence of interest. The components are origin site, then F plasmid origin of replication, then replication E, that is rep protein that specifically binds to the originase to initiate the replication. Then par A and par B are loci that are involved in regular partitioning of F plasmid during cell division. These encodes protein that binds to par C locus and link to the cell membrane. This ensures a regular partitioning of the F plasmid and bacterial artificial chromosomes. The bacterial artificial chromosomes are constructed based on functional fertility plasmid, usually used in transforming and cloning of bacteria, that is E. coli. Supersized inserts capable of holding large pieces of DNA that plasmids can be inserted, and this is originally created from part of the plasmid in some bacteria called as F plasmid. The F plasmid allows bacteria to have sex, that is F plus and F minus are reported to form sex pili, and the transfer of genes takes place between those two bacteria. F could hold up to million base pairs of DNA from another bacterium. The F has origin of replication, and bacteria have a way to control how F is copied. It is a single copy plasmid. Then features are that it can hold up to 350 kilobase pairs, and it can be stable in bacterial genome during the repeated cycle of replication. It holds very large quantity of pieces of DNA and can be cloned into number of times. To study larger genes or the entire viral genome, the bacterial artificial chromosomes is used. Having replication origin, antibiotic resistant genes, cloning sites, and the DNA insert. These are the various parts. Usually in an artificial chromosomes, the first is replication origin, that is required for replication of the clone. Then antibiotic resistant genes, that is to select selectable markers, that is antibiotic resistant genes. If the particular chromosome, if the particular organism is grown in an antibiotics, it will be resistant and it will be able to multiplicate. So in the absence of that antibiotic resistant genes, it cannot multiply and it will be selected against in the antibiotic inoculated medium. So this is very much required for artificial chromosomes. Example of bacterial artificial chromosome is PBAC 1081 developed by Melion et al, and it is called as mini F plasmid PMB 0131. The antibiotics are often added to the media to kill any cells that do not possess the antibiotic resistant selectable marker gene, which is the clone DNA. That is, the remaining cells which has the antibiotic resistant genes will be transfected into the vector, and the cells which do not have the resistant genes cannot replicate itself, and it will die in the antibiotic coated media. The reporter genes. Reporter genes are sometimes called as selective markers, which has different colors like glow or green fluorescent protein in some species of jellyfish. Then color changing gene LAC-Z, blue or white color screening. Then E. coli can be used to either glucose or other sugars, and based on that LAC-Z encodes beta galactosidase and the disaccharide lactose into monosaccharide glucose or galactose. Then a sensitive colorimeter can be used to select the organisms that has the color. Hydrolysis of the colorless analog of lactose by beta galactosidose yields an internal blue product. Then SAC-B gene, a negative selection marker. Here we have leaven sucrose, a gene encoded protein, and which turns the sucrose to leaven, which is toxic to bacteria. And once it is inserted, the bacterial will die, and if the vector carries a DNA insert, the SAC-B is broken and it won't produce the leaven sucrose, and the bacteria will be able to survive with sucrose media. In theory, only the bacteria transected with the vector containing the DNA can grow and form the colonies. As it is the antibiotic marker, here also it is selective marker, and it causes the death of the bacteria that does not have the insert. Then bacterial libraries construction using physical genetic maps. If many model species like Mamasputus, Ziamase, Oryza sativa, in these it has been done for position cloning and physical mapping. Theoretically, it should have several coverage of genome of interest, and the library should have every nucleotide in that genome. Then yeast artificial chromosomes. Yeast artificial chromosomes is a vector and was first described by Murray and Szostak in 1983. It is used to clone DNA fragments larger than 100 kbs, and up to 3000 kbs it can hold, that is 3000 kilo base pairs. So another 1000 has been added, and it is 3 million base pairs can be added in yeast artificial chromosomes. It has an initial circular plasmid which is typically broken into two linear molecules using restriction enzymes. A DNA ligase is used to add the gene of interest between these two linear molecules, and again it will form a single large linear piece of DNA. Then it has different regions like telomere, centromere and origin. Up to this, the various sites were origin, replication enzyme, and then the selective antibiotic marker and the gene of interest, or otherwise the gene of interest which cleaves the SAC-B gene, that is also we studied. Here the telomere, which is located at each chromosome end, protects the linear DNA from degradation by nucleases. Then the centromere, which is the attachment point for spindle fibers, duplicate the chromosome into new daughter cells. Then origin, it is a replication origin which allows the DNA replication machinery to assemble on the DNA, and it is called as autonomously replicating sequence, that is ARS. Then in addition to the above regions, we need another region that we all know, it is the selectable marker A and B, that is bacterial selectable marker and artificial chromosome selectable marker. It is ampicillin resistance marker and recognition site for two restriction enzymes, ECO-R1 and BAM-H1. So in any vector, up to this we have studied, whatever vector, we need first origin, then selectable markers. Then apart from that, the gene of interest is placed, and in case of linear chromosomes, we need the centromere and telomere to avoid sticky ends, which favors cut of the DNA, that is a chromosome fragment. So here we have ECO-R1 site and marker A, marker B, then ampicillin resistance sites, then two telomeres and an origin for yeast artificial vector. So the gene of interest has to be cloned into the site without disturbing these regions, and once it is inserted, it will have the selectable artificial antibiotics regions for selecting ampicillin resistance, and marker A or marker B will be cleaved. Based on that, the selection will be made. If marker A is cleaved, then marker B alone should be there. That can be identified by specific chemical for that particular marker. Then cloning steps involves linearization by restriction digestion. Then recombination DNA has to be transformed into this, that is double orthotrophic mutant like URA, TRP1 yeast strain is used. Then transformers are selected in the minimal media, uracil or tryptophan absent media, as URA3 and tryptophan 1 are present in the double oxotropic mutant. Then here the chromosomes, human chromosomes is used and the vector YAC used, and this is how it is inserted. The human artificial chromosome is inserted in the yeast artificial chromosome and it is replicated, and the one having this particular chromosome alone can replicate in that media. Then yeast artificial chromosomes, it is an eukaryotic organism. Yeast is an eukaryotic organism and hence it can hold more kilobase pairs as compared to the prokaryotes, that is bacteria, phages and plasmids. Then yeast artificial chromosomes, again they are linear chromosomes and they act as shuttle vectors between the bacterial sites and the budding yeast. Genetically engineered chromosomes derived from DNA of the yeast Saccharomyces cerevisiae is ligated to the bacterial plasmid, and the large insert up to 2000 or 3000 kilobase pairs is possible. Stability of the yeast artificial chromosomes also increases with size up to a particular point. The selectable marker is uracil or tryptophan biosynthesis gene, and this is the cloning site is in the middle of SUP4, superior of an Harker alleles of the tyrosine transfer RNA gene. This enables the restoration of normal white color phenotype in otherwise red nonsense mutants. Accordingly, in the international inactivation cloning process, this SUP4 gene is disrupted by the DNA insert, thus removing the suppression of ADA mutation and allowing the phenotypic expression as red color. Minimum features required for existence of yeast artificial chromosome as eukaryotic chromosome in the nucleus. That is, it requires a telomere, centromere, origin, and the sequence required to replicate and maintain the chromosome in the budding yeast. Telomere is to protect the chromosomal end, that is, it should not be sticky ends, it should be blunt end, and it ensures the survival of the chromosomes. The centromere is required for segregation, that is, the spindle fibers can attach only to the centromere. Then origin is required for replication in the yeast, to initiate the replication in the yeast. Then sequence required to replicate and maintain the chromosomes in the budding yeast are also required. After plasmid DNA purification, two distinct digestions are performed. One with BAMH1 and another one is with HIS3. The second digestion of the opening of cloning site is with ICA result, and the linear fragments are produced at left and right arms of the future linear YAC. Selective markers are separated that we already studied. Uracil and tryptophan are the selective markers adjacent to ARS, that is autonomously replicating site, and the centromere 4 on the left arm. Then large DNA fragments which are compatible with the cloning site are inserted into the YACs, and primary transformers can be complemented with URA or tryptophan 1 mutation. Then uses of yeast artificial chromosomes are that it can produce whole DNA libraries of higher organisms like mammalian chromosome sequences necessary for future construction of mammalian artificial chromosomes. Then in Arabidopsis thaliana library, the yeast artificial chromosome genome, the vector PYAC4 suitable for chromosome walking, and it was identified by Grill and Somerville. In RISE genome project, the construction of physical maps in order to represent the entire genome of contiguous sets of overlapping clones, as well as the application in map-based cloning of agronomically important genes, the yeast artificial library of Nippon bear variety was constructed. It has greatly advanced, and the analysis of genomes previously cloned with cosmid vectors are done. The regulation of gene expression of DNA elements present either in the upstream or in the downstream of the eukaryotic chromosomes can be studied. It can be transferred into mouse embryonal stem cells, and subsequent generation in the transgenic mice is also possible. Then difficulties are that they produce chimeric effects, that is, co-ligation of multiple genomic segments into a simple YAC or combination of two or more YAC transformed into the yeast cell that forms a chimeric, that is a mosaic pattern will be there. There may be inserts, rearrangements, sequences with repetition elements and are prone to rearrangements or unclonable elements. Yeast spherophast transformation is relatively inefficient. The next is human artificial chromosomes. This was first given by Harrington et al. In 1997, where the synthesizing is by combining portions of alpha satellite DNA with telomeric region. Very small, roughly 6 to 10 megabases in size, are capable of carrying new genes introduced by human researchers. The components are origin, centromere and telomere. Then chloramphenicol resistance, COS-N, T7 bacteriophage, T7 RNA polymerase driven promoter, and SP6 bacteriophage, SP6 RNA polymerase driven promoter, and restriction enzyme recognition site B and H, shown as BAM H1 and HPA1. So this is the diagram showing the various regions. So this is the replication region and this is the origin region. Then we saw PARA and PARB and PARC, which has to be cloned into various things. Here we have the promoter, various promoter, T7 promoter, SP6 promoter, and 3 and BAM H1 sites, and this is the cloning strip. Next is mammalian artificial chromosomes. Similar to yeast artificial chromosomes, but instead of yeast sequences, they have mammalian or human ones. They can be used to clone very large quantities of DNA fragments, that is more than 100 kilobase pairs and up to 3000 kilobase pairs. In this case, telomeric sequence or multimers of the sequence TTA triple G and commonly used centromeric sequence is composed of another repetitive DNA sequence found at natural centromeres of human chromosomes. And these are also called as alphoid DNA, as they have very large array of tandemly repeating and non-coding DNA. Mostly they are used in clone units of many kilobase pairs. They can carry very large fragments of DNA and again an intact eukaryotic split DNA with exons and introns. After this, the other chromosomes cannot have exons and only introns will be there. It will be spliced off if it is present in the artificially coded target sequence. The MACs are considered to be suitable for gene therapy, where the inserted DNA will be expressed yet stably maintained without affecting the host gene. Then last is P1 derived artificial chromosomes. It was first given by Lono et al. In 1994 and it can hold inserts of 100 to 300 kilobase pairs. It is to avoid the problems of chimerism and instability in cloned DNA. These vectors incorporated features of both P1 and F factor system and can be transferred into E. coli host by electroporation. Several micrograms of cloned DNA can be recovered from 5 to 10 ml of exponential phase of E. coli. There are two types of P1 vectors, that is PNS 583 TET14 AD10 and PAD10 SAC-B2. Both these vectors contain genes of resistance like antibiotic kanamycin and plasmid's origin of replication on LOXP site, the cis-acting site specific recognition signal for the PN recombinase, and a gene that shows resistance to tetracycline which inserts cloned tetracycline resistance. Then the uses of PACs. It is used in genome analysis and mapping of complex plants and animals, isolation of large pieces of DNA rather than small segments. They are also used in phage therapy and in scientific studies focusing on how antibiotics act on a particular bacteria. Then to summarize with, the various artificial chromosomes we studied are bacterial artificial chromosomes, plasmid or plasmid artificial chromosomes, cosmids, and yeast artificial chromosomes, mammalian artificial chromosomes, human artificial chromosomes, or plant artificial chromosomes. They differ in the size of insert, that is the artificial DNA which has to be inserted into it can be of limited size specific for a particular artificial chromosomes. All these things usually have a origin, that is the replication site where it has to origin. Then selectable markers, that is markers can be either antibiotic resistance or some other important conversion like uracil or tryptophan or glucose or galactose encoding enzymes, breaking enzymes. Then these are the selectable markers. And then apart from this, the DNA of interest can be cloned in the region apart from these two regions. Then in linear chromosomes, it requires centromere and telomere for its replication and normal functioning of the chromosomes. Thank you.